Hello, BIO178. Welcome to our Week 7 lab. And in this particular video, I'm going to give us some background information about the particular data set that we're working with today. So overall, when I think about the overall workflow for ecological genomics, which remember is a synonym for molecular ecology. So oftentimes when we're doing these types of studies, the first thing we want to do is identify some kind of environmental variable of interest that we want to investigate. We then want to observe some kind of different phenotypes in response to that environmental difference. And then often once that's established, the next step is to investigate genetic mechanisms that are causing those observed phenotype differences. So just as an example, in a lot of my research, that environmental variable of interest is heat. The phenotype differences are often differences in survival, like the survival curve from my data I showed in the local adaptation lecture. And then once we've established those phenotypic differences, then we can investigate the genetic mechanism. So this is illustrating this process of transcription and translation. So we can look at potential gene expression differences causing those phenotype differences. So in a lot of my research, like I've talked about in some other lectures, and we've actually used some of the data, I've use some of this evidence for the genetic mechanisms. In some of my other research in my lab, we're still at these earlier stages where we're still trying to figure out different phenotypes in response to environmental differences. And so that's actually the data set that we're going to be working with today, and the type of analysis we're doing is at this phenotype level. And specifically, the data set that we're working with today is from the red Abalone Haliotis rufescens. That's the study organism shown here that we're going to be focusing on for this particular lab. Just like the snails, intertidal snails that I've talked about previously, heat stress or high temperatures also affect red abalone. And in particular, it affects both wild red abalone that are found along the coast in the oceans, and also farmed red abalone populations that are shown in this map here. So mortality events occur on abalone farms when there are high temperatures, and in particular, juveniles are the most sensitive to this heat stress, and so that is why we focus on juveniles specifically in my lab. This graphic here on the left is showing that there are some regions along the California coast that get much warmer water temperatures than normal. And for this particular lab, we are specifically focusing on this exposure to high temperatures, but to give you a broader overall picture of the research project in my lab, we are also focusing on this other potential environmental stressor of starvation. So when the abalone experience these warm water temperatures, not only are the abalone themselves exposed to warm water, but it also causes the kelp that they eat to die off. So this picture is illustrating on the left in 2008 versus on the right in 2014. There were huge drastic reductions in kelp forest cover off the California coast, something like 90% reduction in some places because of these warm water temperatures. So these abalone are actually experiencing not only high temperatures, but also starvation. This picture on the right is a sad abalone trying to crawl up a stalk of kelp to eat the fronds of kelp, but there's none left because they're all gone. And why do we care about all of this? Red abalone are economically important, so you may have either heard of people who go abalone fishing, or maybe you have yourself. So usually there's a recreational fishery for red abalone. Sorry, somehow this is set on automatic timer. That's why I need to keep reversing the slide. Generally, there's a recreational fishery for abalone, but that fishery has been closed because the wild populations are not doing well. So that's a loss of about $27 million per year. There's also an aquaculture industry in California where there are farms along the coast, and they farm abalone for sale to restaurants, for instance, for people to eat. This is a picture from one abalone company, Monterey Abalone Company, where they're showing these cages that they grow these abalone in. And so the general research question for this project is how these combined stressors of heat stress and starvation affect survival, and we're also looking at the other phenotype of growth in juvenile red abalone. And I'm showing these images here of 3 students in my lab who are really the ones who have done this research and gathered this data, Danielle, Carissa, and Christina. And I have highlighted in bold that, like I said, overall, this project in my lab, we're looking at heat stress and starvation. For this particular data set we're focusing on for this lab, we are looking at how heat stress affects survival, specifically of juvenile red abalone. And so if we return to this ecological genomics workflow, we've identified now this variable of interest, and this is where we are, is looking at different phenotypes in response to environmental differences. So what we did for this study is we got juvenile abalone from 2 California farms, Monterey Abalone Company, which is not surprisingly in Monterey, and Cultured Abalone Company, which is in Southern California near Santa Barbara in Goleta. For the methods, we got these abalone from the farms, brought them into our lab, kept them in common garden conditions, just like we talked about in the local adaptation lecture. And that's, remember, to erase any past differences in thermal history. They were at these different farms. They may have been experiencing different temperatures to begin with. So we brought them into lab. We actually tag them with bee tags. That's what these little green tags are. We tag each abalone with a unique color and number tag so we can tell them apart when we're measuring growth and that kind of thing. And this pictured on the right is our actual saltwater aquarium system that is in the basement of Sequoia. So we got these individuals, put them in common garden conditions, and then exposed them to a heat stress. And this is just a close-up of the tanks. If you're curious, each of these little guys here is one of the juvenile abalone that were in this study. And what we did is expose them to a 24-day temperature ramp to mimic conditions they would experience in the ocean. And so the heat stress exposure lasted for 24 days. And then the data set that we're working with specifically in this lab is that we then monitored each individual for a 30-day time period after the end of that heat stress. So we basically returned each individual to the normal tank temperature that's supposed to be non-stressful. We monitor them throughout that time period to see which ones survive and which ones die. And then that's what we're working with in this particular lab, is doing an analysis to figure out if the survival of these different individuals from these 2 different farms is significantly different. So again, we're looking at the phenotype level. We're looking at the phenotype level in this lab because in the future, we'll focus on some additional labs that delve further into the molecular side of things and the molecular way we can identify whether local adaptation is occurring. But here for this lab, we'll start first with the phenotype side because that's often the first step in this overall flow of molecular ecology studies, is to first establish if there's any difference at the phenotype level. If there is, then you go and investigate further and figure out what the molecular mechanisms are causing those differences.